So this is where we ended electrodialysis. So this is the second of the membrane separation processes that we need to learn this afternoon. Now, when we speak of electrodialysis, so it's used to transport salt ions from one solution through an ion exchange membrane to another solution. So salt from one solution to another solution under the influence of an applied electric potential difference. That's electrodialysis. Dialysis is actually the transfer of salts from one side of a solution to another solution, or from one solution to another. Electro prefix here was used because of the need to apply electric potential difference for this process to occur. So that's electrodialysis. So in here, you have your feed solution, as you can see in the schematic diagram, being introduced into these frames wherein you have an applied electric potential in here, represented by the anode and the cathode. And then the diluate are those that were allowed to pass through this ion exchange membrane, whereas those that were not are the concentrate, or these are actually those that were not allowed to pass through are the retentate, equivalent to the retentate in the definition of the terms referring to membrane separation processes. The diluate are the permeate, or is the permeate, which is the component which is to be separated from the rest of the components in the feed mixture. The concentrate is comparable to the retentate. Diluate, it's the permeate. To explain the process of electrodialysis better, so I have this video to listen to.
The basic idea is that when you dissolve salt in water, you get positive ion. If you then apply a voltage across that salty water stream, current flows. They are fully flow converters. In electrodialysis, we add a number of alternating current sets of electrodes until either positive or negative. These dynamics are much.
Okay, so a very simple discussion of the use of an electric potential to separate, shall we say, components in water that are supposed to be considered as pollutants, and clear water is separated from such particular components. So the additional mentioned thing there in electrodialysis in the example is their barriers, your membranes in between the two electric potential nodes. They selectively allow only one particular type of ion to pass through, and as such, those particular ions specifically are those that are to be separated from the, shall we say, from the untreated water. Then from the separation of the ions from the untreated water, the product is already your treated water. So that's in the case of electrodialysis being used in the process of water treatment. Now, application involves production of table salt from seawater. So, but class, if you're going to use electrodialysis in the production of table salt from seawater, that's very costly. It could only be air dried, and you already have salt from that. But nonetheless, the salt that we produce just by simple air drying seawater, putting them into compartments, compartmentalizing them, have ununiform sizes, and of course in terms of purity they're not pure. Now it's also used in concentration of brines from reverse osmosis. So from the process of reverse osmosis, further separation process is used, and that particular separation process as mentioned here is your electrodialysis. Now treatment of wastewaters from electroplating. So wastewaters from electroplating processes, of course, have the presence of metal ions. So by properly selecting that particular add-on barrier in our electrolysis process, we separate these ions, these metal ions in wastewaters. These metal ions, you have learned already, is very hazardous to the soil and even to the water itself. Now, In food technology, it's also being used in demineralizing cheese whey. We take out the minerals present in cheese whey to make cheese even taste better. So this is also, it's also using electrolysis. Then production of ultra pure water from semiconductor industry. So this is referring actually to the wastewater, which is a byproduct of the semicon industry. So after the process of ultrafiltration, it can be processed again using electrolysis or electrodialysis. That way, ions present in that particular waters can be separated uh, in the process of producing, take note, ultra pure water. Now, dialysis or liquid permeation is the one that is commonly used by a person whose kidney is not functioning well. In the medical world, it's referred to as hemodialysis. Hemo meaning blood. Dialysis, something to do with liquid permeation. So let's look into what's liquid permeation or dialysis. Now, it is the separation of particles in a liquid on the basis of differences in their ability to pass through a membrane. Again, the membrane is the secret because some will be able to pass through, the others won't be allowed to diffuse through. Small solutes in one liquid phase diffuse readily because of the concentration differences through a porous membrane to the second liquid or the vapor phase. Passage of large particles usually through this membrane is limited or is difficult, or it's being limited by the membrane itself. So that's in the case of dialysis. Now, when you speak of dialysis, we have also two terms that we need to understand properly. So when we speak of the diffusate, it refers to the solvent that is being used in the process of dialysis. The diffusate, the solvent. The dialysate is the feed solution or the one that is being processed. The one or the feed mixture that is to undergo dialysis is dialysate, and the solvent that is to be used in the process of dialysis is the diffusate. So again, you have two new terms here added to your vocabulary in terms of membrane separation. Now, this is a very simple illustration of what happens during a dialysis. So this is your dialysis bag, and inside your dialysis bag is a concentrated solution. Outside this particular dialysis bag, or the solution in which this dialysis bag is actually placed into, contains your diffusate, and that's referred to as the buffer. Now, after a while, so this is at the start of the dialysis, after a while, when your dialysis bag, the concentrated solution specifically in your dialysis bag, has already been equilibrated by your buffer solution, this will happen. So separation of very large from very small molecules is based on the attempt to equilibrate the concentration. So this is concentrated, so the thing is, this will diffuse through the membrane of the bag, and it will transfer the degree or some of the small molecules here or particles inside your concentrated solution inside your dialysis bag will transfer to the buffer solution outside in an attempt to equilibrate both the solution inside your dialysis bag and your buffer solution. So as mentioned in the start of the definition of membrane separation, there is always no sharp separations. As you can see, there are still remaining small particles inside your dialysis bag because we don't actually, and there's no such thing as a complete separation and any separation process that is occurring, naturally occurring. Now, let's discriminate. So I've been using the term diffusion, and there is osmosis, and now you have dialysis. So let's be able to discriminate or differentiate the three terms from each other. That way you will not get confused of which is which. Okay, so let's look at the video. So I hope you were able to differentiate, or you would be able to differentiate a simple diffusion process from osmosis and from dialysis. The thing with diffusion, it does not have a semi-permeable membrane. The two have a semi-permeable membrane. Osmosis is from concentration, less concentrated to high concentrated, while dialysis from high concentration to less concentrated region, just like reverse osmosis. Okay, that's the difference between the three. Now, the application of dialysis includes separation of nickel sulfate from sulfuric acid. This is primarily the process that is being used in mining industries, and hemodialysis, the removal of waste metabolites and excess body water, and restoration of electrolytes balance in the blood. In the case of people who are suffering from kidney failure, the kidney is not able to work properly. So the removal of waste metabolites in the body is like near 0%. Some have only 10% of their kidneys functioning. So the 100% of these waste metabolites have 90% remaining in their bodies, whereas only 10% is uh, able to, let's say, for example, be cleared by the kidney. So the kidney was not able to remove the waste metabolites. In the process of retaining these waste metabolites inside the body also retains water. That is why people who are suffering from kidney problems have big tummies. They retain water in their bodies. That's the reason why major kidney doctors allow or make them measure the water that they drink and measure the urine that they dispense, so that the doctor will know how much of the water they retain in their bodies, because it's already a process that is occurring uh, three ways. They keep the waste metabolites, they have excess body retained in, they have excess water retained in their body, and some of the salts in their bloods are also being depleted in the process. So there has to be restoration also of the electrolytes in the blood. And that is done in the process of hemodialysis, an application of dialysis in the medical world. And I have here a video to show what's happening during hemodialysis. Okay, so that's unit operations applied to uh, taking care of a certain organ in our body that is not anymore functioning well. And that's the hemodialysis, a special branching of the dialysis process. Now we go to pervaporation. There is this prefix per, and you have vaporization. So let's remember these two prefixes, the per and the vaporization. A processing method for separation of mixtures of liquids by partial vaporization through a non-porous or porous membrane. So the reason why you have pervaporation, it's because you have the partial vaporization of your liquids as it passes through your non-porous or porous membrane. So the partial vaporization, meaning some gets vaporized while some doesn't get vaporized in your mixture. That is why pervaporation. But in the next slide, you get to see the real reason behind why the prefix per and then you have vaporization. Now, in the process of pervaporation, your temperature is restricted to below 100 degrees Celsius, meaning there is already vaporization that occurs even at a temperature below 100 degrees Celsius, and that's lesser than the boiling temperature of water. Pervaporation is actually from the term permeation and evaporation. There has to be permeation in the membrane and there has to be partial evaporation of the thing that was permeated through or that has diffused through your membrane. So dense membrane processes are said to be pervaporation processes, and they can be used for selective separation of solvents based on selective sorption and diffusion of one of the components through the membrane. So it's still selected permeation or selective permeation of a component of a mixture in your membrane, and it's a technique whereby the components of the mixture of two liquids are separated by selective permeation through a semi-permeable membrane, the component that passes through the membrane being removed by evaporation or partial vaporization, that is. So that's the reason why you have pervaporation, evaporation, and permeation in one word. And this is used in separating, again, liquid solutions, wherein there has to be two processes that should be happening. The selective permeation of your mixture, feed mixture, that is, component of your feed mixture to your membrane, and those that were allowed to permeate through the membrane will be later on partially vaporized, so that its complete removal from the rest of the mixture in the feed will be effected. So it needs to pass through the membrane, so it needs to permeate, and it needs to vaporize after it has passed through. That is the reason why it's called pervaporation. So you have here a very simple illustration of what pervaporation is. So you have a feed here, and your retentate, of course, the one that was not allowed by your membrane to pass through, lives here, whereas the permeate, the one that was allowed to, the white balls, passes through here, and then the permeate is separated from the rest of the components here in pink balls as a vapor, not anymore as a liquid. And as such, you have the process of pervaporation. In the industry, it's being used, the process that is after a distilling operation. So after distillation, in the condenser, there has to be, or some processes require that there has to be a membrane that will further separate the components that were separated from the heavier boiling components here in your column. So we know that distillation separates the components of a mixture by virtue of the difference in volatilities. So we expect those that are less volatile to be separated from those higher boiling temperature components here at the bottom, the blue ones. These components here that were separated from the higher boiling components will be further separated through the use of this pervaporation. So you have a membrane that will selectively allow some components here that were condensed to pass through and then separate it from the rest of the components in your condensed liquid through vaporization. So you have to partially vaporize it. So now you have the final product here, which is the permeate. Now, so as to the application of pervaporation, so we have the dehydration of ethanol water azeotrope. So we want to separate water, so we need to dehydrate our ethanol water azeotrope. And then azeotrope meaning it has reached a particular state wherein there is no separation already because it has formed already an azeotropic mixture. So the only thing that you get to separate ethanol from water in that case will be through pervaporation. Then we also have removal of water from organic solvents, which uses pervaporation, and removal of organics from water, not just from solvents, but also from water, organics being separated from water. Now we go to microfiltration. Actually, in the next slides, you will have also ultrafiltration, and it's actually an enhancement of the microfiltration process. So it is a low pressure cross-flow membrane process, so cross-flow, for separating colloidal and suspended particles in the range of, so this is how small the particles that needs to be separated, 0.05 to 10 microns. It's a low pressure process since the pressure difference is only 0.5 to 4 bars. And the process is cross-flow, not adjacent flow or parallel flow, but rather cross-flow. The pressure-driven flow through the membrane separates micron-sized particles from fluids which are larger than those in ultrafiltration. So meaning what cannot be processed in microfiltration will now be further processed in ultrafiltration. Because in terms of sizes, ultrafiltration can even allow even smaller particles to pass through the membrane. Now, this particular process is used in the separation of heterogeneous mixture. Heterogeneous in the sense that the sizes of the particles are not the same. Now, in microfiltration, so you have the application of the pressure in this side, wherein you have a mixture of small and large particles. And in the application of pressure, you have this particular membrane that will only allow a certain particle size to pass through. So only the smaller particles will pass through the application of this force here, pressure that is, and the bigger particles, of course, will be retained on this side. So this is microfiltration. This is also being used in water treatment. Now, there are techniques, actually, two techniques that are being used in microfiltration process. So though it's mentioned already that the process is cross-flow, so as you can see here, cross-flow filtration, but we can also have what we call as a dead-end filtration. So it could be dead-end filtration in this case, and it could be cross-flow filtration. But oftentimes, what is being used is dead-end filtration, because in here you have all the feed mixture being processed in one direction through this membrane here, whereas in here it's cross-flow. The process of applying your feed mixture is in this direction, whereas the permeate is flowing from your membrane in this direction. So that is why it's called cross-flow. Now, you have this video on the process of microfiltration. What actually happens during microfiltration?
During microfiltration, water is forced through lots of vessels, with each vessel containing thousands of hollow fibers. The outer wall of each fiber is made up of tiny pores, only 0.4 microns in size, or about 300 times smaller than the width of a human hair. The fibers filter out microscopic particles such as cells, protozoan cells, bacteria, and even viruses. Microfiltration vessels are backwashed every 30 minutes to remove any buildup of particles on the membrane walls. The back wash water is returned to the filtering for reprocessing. This helps the plant recover a higher amount of pure water.
So it's just like filtration that uses very, very small, 300 times smaller than a single strand of our hair. So the membrane is a bunch of this hair thin, or even thinner than the hair, strands that filters out yeast, as mentioned, bacteria, and even viruses from our water. So that's microfiltration. The process is very simple. It's capitalizing on the difference in the size of the particles that needs to be separated from water that is. Now, application, separation of bacteria, paint, pigment, yeast seals from solutions. Sterilization of liquids, gases, and parenteral drugs. Clarification and biological stabilization of beverages, bacterial cell harvest, and purification of antibiotics. So look at its very important application, and recovery of mammalian cells from cell culture broth. So this is using, all of these processes are using microfiltration in the process of separation. So I have 10 minutes remaining, so I need to finish. Now, ultrafiltration is also pressure-driven, and pressure is used to obtain the separation of molecules through a semi-permeable polymeric membrane. The membrane discriminates on the basis of molecular size, just like microfiltration, even shape or chemical structure, and separates relatively high molecular weight solutes, such as proteins, polymers, and colloidal materials, such as minerals. A pressure-driven process similar to reverse osmosis and microfiltration using, take note, asymmetric membranes, but that are significantly more porous. Okay, this is the last video in this slide. Sorry. So if we think that microfiltration already gives us safe drinking water, then we can have ultra safe water that is, of course, can process as small as 0.01 micron, with small sizes of pores as small as 0.01 micron. This is used in the pre-concentration of milk before making cheese. So it's used actually in majority in the food industry, clarification of fruit juice, purification of recombinant proteins and DNA, antigens and antibiotics from clarified cell broth, and color removal from craft black liquor in paper making. So these are applications of ultrafiltration of processes. So this is, again, how we distinguish the different processes. So you have in water purification as an example, the first part could be microfiltration, followed by ultrafiltration, Then some are even using nanofiltration, and after that we have reverse osmosis. The final product will be ultra safe drinking water. So this could be a complete set of processing for our wastewater, very dirty wastewater that would be now considered as very safe to drink wastewater, originally wastewater. These are the particles that will be subsequently removed in the process in these different um, membrane separation processes. Now, gas permeation, this is the second to the last topic here, uses a dense polymer membrane which is non-porous, such as rubber or polyamide. Now, the solid gas dissolves first in the membrane and then diffuses in the solid to the other gas phase. The feed is at a high pressure and usually contains some low molecular mass species, now typically less than 50 kilogram per kilomole, to be separated from higher molecular mass species. Now, pressure gradient here that is needed for the process is 20 to 40 bar. So take note of this special type of barrier or membrane that is being used in gas permeation. It's rubber or polyamide. Now, gas permeation here, in this case, you'll have a component of a gas mixture having component A, and this is component B. This is where your feed composed of mixture of A and B entering your gas permeation equipment, and then through low pressure you are separating. You could see here you are separating the rich in A component retentate, and this B, the smaller molecules, is your permeate. So separating the two by virtue of this particular membrane, which is labeled here as a zeolite membrane. If you recall, the membrane is polymeric in nature. So in here, the membrane that is typically used is that of a zeolite. Okay, last, I think this is the last on gas permeation. So that's how gas permeation works in the case of the removal of CO2 from a gas stream. It's just like you're separating a gas mixture and a component is allowed to permeate through the membrane, whereas the other one is not. So you are separating already the components of your gas mixture by means of gas permeation. Now, these are the application of gas permeation. So separation of CO2 or hydrogen from methane, the separation of uranium isotopes, adjustment of the H2 to carbon monoxide ratio in a synthesis gas, the separation of air into nitrogen and oxygen enriched stream, the recovery of helium from natural gas using a fluorocarbon polymer, and the recovery of methane from biogas. But these are just one of the many applications of gas permeation. Now, gas diffusion in a porous solid, on the other hand, so in here you have a microporous solid membrane separating the two gas phases present in both sides of the membrane. The rates of molecular diffusion of the various gas molecules depend on the pore size and the molecular weights of the gases. Separation of the gaseous components is achieved using the selective properties of the membrane. When the pores are much smaller than the mean free path in the gas phase, about 1000 angstrom at standard conditions, the gases diffuse independently by Knudsen diffusion. And what is this Knudsen diffusion? So in comparison, this is Knudsen diffusion here, wherein your gas molecules passes through your membrane in all direction. You could see this. It bounces off or it collides on the surface of your membrane, and the permeate is separated in this manner. Whereas this one is molecular sieving, a simple molecular sieving wherein the smaller molecules are allowed to pass through, whereas this one is solution diffusion. The solution allows only to diffuse the smaller particles, just like in molecular sieving. In Knudsen diffusion, there is this application of a pressure differential. That way, your gas mixture will pass through the membranes. So these are all your membranes. The rest that doesn't pass through remains on the other side, which is your retentate. The rest that passes through the membrane are your permeate. Now, if I will be cut through in this particular slide class, I will just let you read the rest of the slides here because they are just easy to follow through. Okay, so these are the application of gaseous diffusion in porous membranes. So production of enriched uranium by forcing gaseous uranium hex